Yeah, so today we have Dr. Matt Kaeberlein here, and he is a pretty important researcher in the field of anti-aging, and so we just wanted to bring him here. So we're hosting this conversation on behalf of Reddit/r/longevity, and also for the Health Span Discord forum. And so I have some questions, but first I'd like you to just, I guess, introduce yourself and talk a little bit about what you do. Yeah.
Sure. So my name is Matt Kaeberlein. I am a professor of laboratory medicine and pathology at the University of Washington. I have been studying the biology of aging since I was a graduate student. I got my start working on aging in a single-celled organism in budding yeast as a grad student, and then as a postdoc, got interested in the question of to what extent are basic mechanisms of aging conserved, and so started working in C. elegans, which is a nematode worm. And then since I've started my own lab, we've branched out to do studies of aging in mice. We have a pretty large project studying aging in companion dogs, in pet dogs. And then I've gotten very interested in translational approaches to actually apply some of what we've learned about aging biology to improve health and longevity in people.
So the current research, the dog project, how's that going? And I guess the first question is, when can we expect the first data from the dog aging project?
Sure. So the dog aging project, just to give a little bit of background, is a large study with really two complementary goals. The first goal is to really understand what are the most important genetic and environmental factors that influence healthy aging in pet dogs. And then the second goal is to do something about it, to ask whether or not we can actually apply what we've learned from preclinical studies, from laboratory studies of aging, to actually increase healthy lifespan in pet dogs. And so the largest component of the dog aging project is what we call a longitudinal study of aging. So it's really an observational study where we are following pet dogs living with their owners, going about their normal lives over a period of 5 to 10 years, maybe even longer, with the goal there of really understanding what are the mechanisms of aging in dogs. And so that study is really just kicking off. So we have enrolled several thousand people now in what we call the Dog Aging Project PAC. So these are owners who have nominated their dogs to participate in the study. They are providing data on their dogs, both through extensive surveys about their dogs' health history, environment, diet that they eat, basically all of the different factors that go into the sort of overall environment that dog is experiencing. They also are providing us with veterinary records. So we have a electronic medical record for those dogs. And then over the next 5 to 10 years, as those dogs go throughout a large fraction of their normal lives, we'll learn a lot about the health outcomes that those individual dogs experience. So we anticipate sending out invitations to all of the 80,000 plus people who have asked to participate over the next couple of months. We're hoping that number gets up over 100,000. So I think it's quite likely that within the next year, this will be the largest citizen science project in the world. And it's really aimed at studying the biology of aging in dogs. A subset of the dogs that join the Dog Aging Project PAC will be studied in more detail. So there's a group of dogs, which we call the foundation group, where those dogs will get their genomes sequenced. So we'll have genetic information on those dogs. We hope that will be above 10,000. We're funded to do that for about 8,500 dogs right now. And then there's what we call the precision group. And those are the dogs where we get the most detailed information. So we'll get annual information on their blood metabolites, blood chemistry, fecal microbiome, and epigenome. And so the idea there is to really take a systems biology approach in that sort of high resolution group of dogs to really understand in detail, what are some of the molecular markers that are predictive of healthy aging. And then there's a smaller part of the dog aging project, at least in terms of number of dogs, which is really focused on the second goal of trying to increase healthy longevity in dogs. That's the Triad clinical trial. Triad stands for Test of Rapamycin in Aging Dogs. That's a double-blind, placebo-controlled, randomized clinical trial designed to answer the question, does rapamycin slow aging in dogs? And so that is actually our third clinical trial in this series. It's the largest. The first two were really safety studies. So those two studies are done, and the Triad clinical trial will enroll a few dogs, probably fourth quarter of this year, and then we'll really kick off and try to get all 500 dogs into the study next year. So probably starting in February is when we'll really roll out the large scale enrollment for that clinical trial.
Okay. So for the epigenetics, do you mean just methylation or also like acetylation or?
Yeah, so at this point, we're looking at specifically at methylation, and that is really based on a growing body of evidence in the field that that you can develop these epigenetic clocks based on methylation that are very predictive of chronological age. And there's some reason to believe that you can also develop clocks that are predictive of biological age. And in fact, a recent paper from Elaine Ostrander and Trey Eidecker did this in dogs. And they actually found this sort of interesting observation when they tried to compare the dog epigenetic clocks to the human epigenetic clocks, which is that dogs age more rapidly than people do, but it's not sort of a linear relationship. In other words, we're all used to this idea that 7 dog years equals one human, right? And what they found is that, at least based on the epigenetic clocks, that it's really a much higher rate of aging in dogs early on in life, like maybe 14 years to 1 human year. But then once the dogs get to be 2-3 years old, then that rate of the relative rate of aging, again, at least at the epigenetic level, sort of slows down, and then it goes down to maybe 3 dog years for every human year. So that's really interesting. Again, I mean, we always have to be a little bit careful not to assume that these epigenetic clocks are really reporting on biological age. That, I think, is still a little bit unknown. But there's reason to believe that you can certainly get predictors of health during aging from these epigenetic clocks. And so we're very interested in asking, as we build these clocks in our precision cohort, can we make predictions about the health outcomes and lifespan of individual dogs that then we'll be able to see later on and ask whether those predictions actually worked out the way that we predicted. Another interesting question is, are there breed-specific epigenetic clocks that monitor aging in dogs? So, for example, just in a general sense, big dogs age faster than small dogs.
Right.
Certainly for lifespan, the lifespan of a 150-pound dog is going to be about maybe half that of a 30-pound dog. And it seems to be accelerated aging, not just a difference in lifespan, in the sense that big dogs develop, all of the same diseases and functional declines of aging that we see in small dogs, but they seem to develop, most of them at least, more rapidly. And so you might predict that if you were to look at the epigenetic clock for chronological aging that across all dogs and apply that to breeds, the question is, can we see this putative accelerated aging in large dogs versus small dogs, or in certain breeds of dogs? There's a lot of interesting biology to dig into with these epigenetic clocks.
That's kind of counterintuitive too, because typically larger animals have longer lifespans. So it's like, but it's reversed in this situation.
So it is from that perspective, although, and this is something that a lot of people get confused about, including some people in the field. So when you look across species, what you just said is absolutely true, that bigger species tend to live longer than smaller species. There are obviously some outliers, but in a general sense, there's a really good correlation between body size and at least maximum lifespan across many different species. But when you actually look within species, that that correlation is usually reversed in the sense that when you focus on a single species, smaller individuals tend to live longer than larger individuals. And in dogs, I think we're just able to see that because there is such a larger degree of variation in body size. So that relationship is just more pronounced in dogs than it might be in people or in mice or in other animals that you might look at. But even in other animals, and again, the data is not as good as it is in dogs, the data that exists suggests that relationship is probably also conserved across animals, where within a species, smaller individuals tend to live longer and appear to age more slowly.
Is there a stronger correlation within a species or of reproductive age with longevity?
Right. So age at which the species reaches sexual maturation certainly is correlated with longevity. I don't know how carefully it's been looked at in terms of reproductive senescence. So, you know, there's the age at which
the end of fertility. Yeah.
And then there's how long can that species reproduce before it becomes sterile. And so I don't think that's been really looked at as carefully, but certainly it makes evolutionary sense that species lifespan would have to be at least somewhat correlated with age of sexual maturation, because obviously the species has to live to the point where it is able to reproduce in order to be evolutionarily successful. I think it is a really interesting question, you know, particularly in humans, for example. There is some evidence that I'm aware of that age of menopause in women is probably also correlated with lifespan. So in other words, later age of menopause, those individuals probably tend to also live a little bit longer. And that makes intuitive sense. I'm not sure how carefully that's been looked at in terms of some of these molecular markers like the epigenetic clock that we think might be surrogate measures of biological age. It's a really interesting question, actually, that is addressable. I just don't know if anybody has done it yet.
Mm. Are you looking at telomeres for your dogs at all, or?
So we haven't yet developed any formal plans to look at telomeres. So we are in the precision cohort, we are going to be collecting PBMCs, blood cells that we can look at. So in principle, we could do measurements of telomeres from blood from the precision cohort, but we haven't really figured out the right partners to do that with at this point. So this is one of the challenges, as you can probably imagine. I mean, just in the course of this discussion, we could probably come up with 25 things that would be great to measure, right? And you just really, for a variety of reasons, you can't do it all, money being one. But also, when you actually start to think about the logistics of having thousands of dogs come into hundreds, maybe thousands of different veterinary clinics around the United States, how you actually collect those samples, get them shipped to the right place in a way that is consistent with then being able to do some of the downstream studies you might want to do, like measuring telomeres. It just gets very hard to do a lot of the sophisticated kinds of things that you could imagine doing. So we have tried to structure our studies, first of all, around the principle that we want to make it as easy for the owners and the dogs as possible to participate. So really nothing that is invasive or would cause undue stress on the dogs. And then also assays where the handling of the samples is going to be consistent with getting good quality data out of those assays. So for example, most private veterinary clinics don't have liquid nitrogen on site. So if the assay you wanted to perform required that the samples be put on liquid nitrogen right away, we just wouldn't really be able to do that and get good quality data out. So those are some of the considerations that we've made when we've tried to think about what can we measure, what should we measure, what do we think we can get the best quality data that's also going to be informative from.
I'm going to bring up a couple questions from the forum that have been dropped here. One person asked if you could speak to the nature of the fraudulent research that's occurring in the longevity field. People who put out papers that are just really bad and they continue to get funding and stuff like that.
Yeah, so the first thing I would say is I wouldn't use the word fraudulent. So I think that fraud in science is actually very rare. I think that it's not common that people intentionally falsify or make up data and publish it. It certainly does happen, but I think the vast majority of the results in the field that haven't been reproducible are more likely due to a couple of factors. One factor is that some of these experiments are just very sensitive to the way that they're done, that different labs do things differently and don't always reproduce each other's data. So some of it is, part of the normal way that science works. And figuring out which results are robust and reproducible. Certainly some of it is due to sloppy science. And I think that the aging field has its share of sloppy science, but every field does. And, some of that is, this is my personal feeling, some of that is just driven by the fact that there are a couple of different, I think, ways that people view science. I am very much of the school of thought that our goal as scientists is to disprove our models. We develop a model, we develop hypotheses, and we try to break those models because our models are wrong, right? Our models are sort of imperfect descriptions of reality based on what we know at the time. So my personal view is that scientists, and this doesn't just apply to the aging field, this applies to science in general, scientists should always go in recognizing that their model is imperfect, and we should design experiments that try to break the model. There are absolutely people in the aging field and in other areas of science that take a different view, that they think that their model is right and they believe that they get results to their experiments that don't fit the model and they ignore those results. And so my personal opinion is that most of the controversies in the aging field stem from that, from people ignoring data that didn't fit their model. A byproduct of that is that allows them more often than not to publish high profile papers because most of the high profile journals like very simple stories, right, that don't have complicated data. And so that feeds into mistakes in the literature that persist for a while before they get worked out. And I think there are good examples of that in the aging field. I'm not going to name them because I don't feel like I need to call people out in this forum. But I think that my personal view is that really feeds into a lot of the controversies in the field. It's the difference between believing that you know the answer and getting data that fits it, which I think is a mistake, versus recognizing that you don't know the answer. You might know part of the answer, but we should try to do experiments to figure out where our model is wrong. And that's the approach that I take always in my science.
Do you feel like there's any particular areas of aging research that are neglected too much?
That's a good question. I haven't really thought about it in that context. So I think I wouldn't say it's neglected. Let me answer the question this way. I think that one of the major challenges for the field going forward is figuring out how we take the next step. How do we take the answers that we've gotten from the basic science and really apply that directly in the real world? So I think it's still, I think one of the outstanding questions in the field is how much of what we've learned in laboratory animals will really apply to aging in people living in the natural environment, or in the case of the Dog Aging Project, to dogs living in the human environment and their companion dogs living in their natural environment. It's a real unknown. And I think we need to be honest about that. I think there are good reasons to think that much of what we've learned from laboratory animals will be true in the real world, but there's also good reasons to think that some of it won't. And so I think figuring that out is an area that not a lot of attention has been paid to, where I think that it's where the field's going. There's no question that there are lots of people interested in translational geroscience, as I call it. So taking what we've learned about aging from the laboratory and trying to use that information to improve health and longevity in people or in companion dogs. So that's a direction the field is going, but there aren't a lot of examples of this being done in a rigorous way yet. And I think that there are people thinking about that, and I'm not blaming anybody. It's a really hard problem. but I think that's where the field should continue to pay more attention. And I think we definitely see examples of where it's clearly not being done in a rigorous way. And I'm uncertain as to how problematic that's going to be for the field going forward. So just as an example, we all know that there are a variety of different unregulated natural product mixtures and other treatments that are available that the FDA is not regulating. We have no reason to believe that any of them really work in people. And there's no incentive for the people who are selling these things to actually do rigorous clinical trials to find out if they work.
Right.
That's unfortunate, and in my view, it has the potential to create problems for the field down the road. I don't know how much of a problem it's going to create, but I think it's clear that, you know, if something went dramatically wrong with one of these things, that could be very problematic.
Have you heard of any research that aims to translate multiple different research observations into, I guess, convergent therapies?
So, yeah, so I mean, certainly people are talking about that. Again, that's, you know, the real challenge is how do you do that in a rigorous way in the context of the current, you know, regulatory environment, right? So,
because that is a significant barrier. Absolutely.
So you know, there are really a couple of major approaches that people can take, right? You can try to develop a product based on natural products that are not FDA regulated and start selling it. Or you can try to do it through at least in the United States, through the FDA regulatory path. There are real challenges with doing it through the FDA regulatory path. We all know that that's an extremely expensive and complicated and time-consuming process for a sort of traditional pharmaceutical approach. When you're talking about aging, where aging itself is not a disease or an indication, meaning you can't get FDA approval for aging, at least not yet, that obviously adds another level of complexity. So certainly there are people talking about combinatorial approaches. That's another area where I would say the basic research isn't really all that well developed yet. So there are a few labs that are really trying to test this idea that you can take multiple interventions that there's at least some reason to believe act through different pathways and combine them and ask whether or not you get synergistic or additive effects for health and longevity. But there aren't very many labs doing that, and the data there, you know, is pretty small data set. Certainly there are also groups that are thinking about doing this in humans. I don't know of anybody who's really seriously thought about doing this through the FDA regulatory process. You know, you got to start with one thing before you try to combine things. But certainly on the natural product side, there are groups who are talking about doing this. Some of them may already be to market. The one thing I would add is I have a little bit of hope that some of the groups who are pursuing the non-regulated path are in fact actually starting to look at some of these clocks, right? So the epigenetic clocks, the blood clocks, other clocks that have been developed that might be reflecting biological age. So they're at least starting to ask the question, you know, in people taking this product, do we see changes in these clocks that are consistent with the product actually having the effect that we hope it has? So I didn't mean to give the impression that everybody in this natural product space is just out to make money. But those examples of groups that I know of, at least, that are really starting to try to test their products are the exception rather than the rule. Yeah. So most of the groups that I know that are selling, you know, quote unquote anti-aging products are not testing their products at all. Or if they are, they're testing them in ways that don't really tell us anything other than it allows them to send out a press release saying that they, you know, did a clinical trial for some endpoint that's meaningless.
Yeah. Another question was, can you speak about COVID-19 and how current wave of people recognizing aging, senescence, and mortality, and how's that
role in COVID-19? Yeah, so I mean, it's an interesting. I mean, it's obviously tragic, right? And it's something that's affecting all of our lives.
Right.
But it's also an interesting time for the aging field because what you said is exactly right, that it's absolutely clear that COVID-19, at least severe complication and death from COVID-19, is as much an age-related disease as Alzheimer's disease and heart disease and diabetes, in the sense that your risk of getting extremely sick or dying from COVID-19 once you're infected shows almost identical age-related kinetics as all those other diseases that we recognize as diseases of aging. And to those of us who studied the biology of aging, that makes perfect sense. I mean, that was, one of the first things that I thought when it became clear that this was, more than just a weak version of influenza, was that, this is going to be very much a disease of the elderly. And in fact, I live near Seattle, and we were sort of the first hotspot in the United States that got recognized. And it was an outbreak at a nursing home where, many of the people there got sick and died from COVID-19. And so that fit perfectly with exactly what we would expect, that this is a disease that tends to most severely affect the elderly. So then, the question is, there are a couple of questions. One is, how do we actually get people to recognize the important role that the biology of aging is playing in this? So the observation is clear. I don't think anybody would argue with the observation that if you're older, you're more likely to get sick and die from COVID-19. But how do we get other scientists and hopefully some portion of the general public to understand that there is a real biology here that's causing that, and that if we can actually modulate that biology, we have the potential to have a much more significant impact on these severe outcomes and deaths from COVID-19 than the strategies that almost everybody is talking about. So that's a challenge. I think that there have been some successes, but overall, I am not really sure that this message has gotten out as broadly as it needs to. And one of the reasons why I would say that is that if you look at the federal funding for COVID-19, there were several $100 million that the federal government put towards research on COVID-19. Absolutely none of that money went to the National Institute on Aging, which is the institute that funds aging research. So despite the fact that age is the greatest risk factor of dying from COVID-19, $0 out of the hundreds of millions of federal dollars for research on COVID-19 actually went to address that problem. So that tells me that at least at the level of the policymakers and the administrators at NIH, they haven't gotten the message that they really need to start paying attention to the important role that aging biology plays in diseases like COVID-19, and also in diseases like influenza, which we just sort of accept will kill a few tens of millions of people every year. Again, age is the greatest risk factor for dying from influenza. And there's good reason to believe that we have opportunities to prevent those severe outcomes and deaths by directly targeting the biological mechanisms of aging.
I'm going to switch a little bit here. What can the longevity community do to help speed up research, either your research or your research in general?
So I'm not sure how to answer that question, because I'm not sure what the person means by the longevity community.
Oh, okay.
So, I mean, I think, those of us who are in the longevity community on the research side, we're plugging away, working as hard as we can to move the field forward. I think, there are obviously anybody who works in academic science and has to write grants all the time will note that it would be great if there was more money for research in this area. And that is true. It's also sort of obvious, but that is certainly the biggest bottleneck, I would say, to more rapid progress in the area of biological aging. There is also a component of messaging, and I think that's an area that I've started to try to become more active in, and several of my colleagues as well, educating the policymakers, other scientists, the general public about the great advances, I would say, that have been made in understanding the biology of aging, the real opportunities that are there for taking some of these discoveries and applying them in human medicine and in veterinary medicine. So there is a component of getting the word out there. And so I think that's an area where we can do better. So I would say, funding is desperately important for moving the field forward, and also messaging and getting the message out there. And obviously, if we're successful at getting the message out there, that has the potential to lead to additional funding for the field.
Here's a very specific question. Hopefully I read this correctly. In considering the differences between RTB101 and RAD001, as you discussed in your recent paper of the Restore Bio failure, as well as Manic's attribution of the failure to the FDA changing the primary endpoint to a subjective measure, if the company posts positive results in its COVID-19 trial, would it be appropriate for the geroscience hypothesis to take credit, or pragmatically would doing so be politically favorable?
There's a lot to unpack there. So
yeah.
let me spend about 30 seconds just giving background to the listeners who might not be in on this story.
Right.
So rapamycin, of course, is a drug that is well studied in the aging field. It's probably the gold standard drug for increasing lifespan and slowing aging across lots of different organisms. There was a study from Joan Manick, who is at Restore Bio, which is a company. There were two studies actually over the past decade showing that if you take a derivative of rapamycin, which is the RAD001 that the viewer mentioned, you take that derivative of rapamycin, and I think for all intents and purposes, we can just think of it as the same as rapamycin, and you give it to healthy older people for 6 weeks, they have a better flu vaccine response, and they actually have fewer respiratory tract infections over the subsequent year. And so that was consistent with the mouse data suggesting that rapamycin or a rapamycin derivative can rejuvenate immune function in people, which is obviously super exciting, very relevant for the discussion we just had about COVID-19 and influenza. Okay, so Restore Bio also was working with another drug, this RTB101, which is not a derivative of rapamycin, and it's actually a class of molecule called a dual kinase inhibitor. So it inhibits the same protein that rapamycin inhibits, which is called TOR. So it has that similar effect to rapamycin, but it also does other things. So in some ways, you can think of it as a less specific version of rapamycin that does lots of other things. As far as I know, RTB101 has never been tested for effects on lifespan in mice or other model organisms in the lab. So there's a large body of data for rapamycin that it slows aging. No data that I know of, again, really suggesting that RTB101 slows aging, but you might think it does because it overlaps with rapamycin. So Restore Bio took RTB101 forward into what's called a phase 3 clinical trial to try to get FDA approval for these immune rejuvenating effects. And their clinical trial failed. And your listener's absolutely right that Dr. Manick has suggested that failure of the phase 3 clinical trial was because the FDA changed the endpoints. I haven't seen the data, so I can't really comment on that. But I think the failure of that clinical trial did, in my mind, raise the question of, would it have been better to move forward in these large-scale, very important human studies with a drug like rapamycin or RAD001, where we have good reason to believe that it affects aging, rather than RTB101, where we don't really know whether it affects aging. So I guess that was more than 30 seconds, but that's the background. So then the question is, so what has happened with COVID-19 is Restore Bio has moved forward with a second phase 3 clinical trial in patients in nursing homes to ask the question, does RTB101 protect those people against likelihood of severe outcome or death from COVID-19? I haven't talked to Dr. Manick or anybody at Restore Bio, so I have no knowledge of the status of that trial or what the data looks like. But the question is, if RTB101 works, should the field of geroscience take credit for that? And as I've just suggested, it would be a little bit intellectually dishonest for us to say that this is proof that the geroscience hypothesis works, right? Because we don't know whether RTB101 slows aging in mice or other organisms. On the other hand, because the target of RTB101 overlaps with rapamycin, you know, it certainly is consistent with the geroscience hypothesis, I would say. I would just like to see somebody go back and do the mouse studies, and I would love to do these studies, to ask, does RTB101 have the same effects in mice on aging and a variety of age-related functional measures that rapamycin does? We just don't know the answer to that yet. So it's a little bit unclear how similar rapamycin and RTB101 are at the level of their effects on aging. Having said that, you know, I am extremely hopeful that the Restore Bio trial of RTB101 for COVID-19 works, because that would be really important, obviously, to have a drug that might affect aging, that appears to rejuvenate immune function and could help older people who are at the highest risk of severe consequences from COVID-19.
I have a personal question. Do you think that the force of natural selection is pushing us towards longer lifespans or for shorter lifespans?
So I'm not even going to try to answer that question for
Oh.
humans, because humans have, you know, I mean, we're not naturally selecting anymore. We're in
Right.
this state of unnatural selection, I would say. And human lifespan is so artificially manipulated already that, you know, it's kind of, there's just no way I can make an intelligent comment on that, I don't think. I may not make an intelligent comment on the question at all, but I don't think I can do it for humans. So if you think more generally about most species, it is my opinion, and I think most people in the field would agree with this, that natural selection is largely acting independently of aging. In other words, there's not a lot of reason to believe in most species that natural selection is acting at the level of lifespan, or even at the level of functional declines and diseases of aging. Instead, it seems pretty clear to me, at least, that natural selection is largely acting on what you would think it would be acting on, which is rate of sexual maturation, and then once you're sexually mature, fecundity, so rate of reproduction. But that's all in the context of the specific environment that the individual is existing in. And so the way I think about this is, you know, there are fundamental decisions that all organisms have to make, right, in terms of survival. And one of the most basic is, when is it a good time to reproduce? And an important consideration, you know, at the organismal level of whether you should have babies is, what's the environment like? Is the environment appropriate for the survival of your offspring? And so I think all organisms have had to evolve to sense the environment and kind of make this fundamental decision: grow and reproduce, or if the environment is unfavorable, shut down growth and reproduction, become stress resistant to withstand that environment until things become favorable again. And when you think about the environmental cues that are likely to be important for making that decision, one of the most important is going to be food. So is there enough food around for your babies to grow and reach sexual maturation themselves, to pass their genes on to the next generation? So from a selective standpoint, I think this decision is really important. And I think nutrient status is probably one of the most important environmental cues for making that decision. And so that ties in really well with what we have learned about the kinds of environmental interventions and the cellular pathways that affect aging. Most, if not all of them, are involved in making this decision and responding appropriately. So we talked a little bit about TOR. That's the target of rapamycin, the protein that rapamycin inhibits. TOR is a really good example. Its fundamental job in the cell is to sense nutrient status and help the cell make the right decision. Should it grow and reproduce, or should it shut down and become stress resistant? And when nutrient levels are low, TOR shuts down and the cell becomes long-lived. So the whole reason why I'm going through this is that I think it's important to consider that natural selection is unlikely to be acting on these genes like TOR because it's selecting for the role that those genes play in lifespan. I think it's more likely that it's acting on these genes because of this decision-making process that these genes have to make. And it's just a byproduct of that decision that they affect, broadly speaking, stress resistance pathways and growth-promoting pathways that are associated with lifespan and rate of aging.
I have a comment on that further question. What of the grandmother hypothesis for K-selected species?
Yeah. So I mean, so again, this is the idea that that at least in very long-lived species, where you might have multiple generations still alive all at the same time, that the grandmother, so to speak, provides value to the mother and the granddaughter in terms of the wisdom that gets passed on, and perhaps even the sort of capacity for care that the grandmother can provide to support the mother, and thereby enhance the selective advantage of that granddaughter to reach sexual maturation and pass her genes on. So I think that that's absolutely valid, and intuitively, it seems likely that that has some role in certain species. I haven't personally seen data that convinces me that this is a major component of natural selection. So I'm not at all arguing that it doesn't contribute, but I think it's probably a minor contributing factor relative to these other forces, such as the one I was just describing about making the appropriate decision about growth versus no growth. But certainly you can make an argument that there is some contribution to kin selection and things like that, at least for certain species.
So maybe we should be concerned about which aspects of our shared environments around the world contribute to these genetic stress responses.
Yeah, I think that's absolutely right, and this is an area that, you know, there is some work going on, but not a lot, and that's actually something that I think, you know, we'll be able to provide some insights into from the Dog Aging Project, because dogs age so much more rapidly than people do. We'll actually be able to identify some of these environmental correlates with health and longevity in a reasonable time frame. Obviously, because this is a longitudinal study in companion dogs, there'll only be correlations. So we won't be able to say, you know, with a high degree of certainty that this particular environmental factor is causing this positive or negative outcome in the context of aging. But it will certainly be hypothesis developing, and should set the stage for some more mechanistic targeted studies that can be done then in a more controlled environment. I think, you know, a real obvious example here is the role of air pollution in life expectancy and potentially in rate of aging. Tuck Finch at USC has done some brilliant work in this area that is clearly a major environmental concern in many parts of the world, especially the parts that are most densely populated. And so that's a real obvious case where, you know, an environmental factor could have a pretty significant effect, certainly on life expectancy and potentially on the rate of aging in individuals that are exposed to high levels of air pollution.
How many, was it populations or cohorts does your study have?
So right now, in the longitudinal study, so there's the triad clinical trial, which is sort of its separate group. In the longitudinal study, there are 4 distinct groups at this point. We're trying not to use the word cohort, in part because of, you know, this idea of messaging to the general public. So we call them groups. There's the pack, which is the largest group, and again, that basically, to be in the pack, all an owner has to do is have a dog, nominate the dog, complete the surveys, and send us their veterinary medical records. In fact, they don't even have to send us their veterinary medical records, but we really would like them to, because that's where, you know, a lot of the power is going to come from. So that's open to everybody. And then there are the dedicated groups where, at least for now, because of budgetary reasons, we're limited in the number of dogs we can enroll into each of those groups. There's the foundation, which is the approximately 10,000 dogs that will have their genome sequenced. So that's where we'll get the genetic information. And then there's the precision, which we talked about, which is the high resolution omics group. And we'll see those dogs annually. And then there's another group, which I haven't talked about, that we call the centenarian group. And so these are dogs that appear to live exceptionally long for their body size. So as we already discussed, big dogs age more rapidly than small dogs. So for example, if you had a 21-year-old German Shepherd, that would be an exceptionally long-lived German Shepherd. And so we're recruiting a few hundred centenarian dogs to try to see if we can identify correlates in either their genomes or their environment that are associated with their exceptional longevity.
I'm a little bit confused. You said you would treat some dogs with rapamycin?
Yeah, so that's the triad clinical trial. So that's a different group, and that is a group of 500 dogs. Half of them will get placebo, half of them will receive rapamycin. That's a 3-year clinical trial. So the dogs who are enrolled into that study will be in the study for 3 years. They come to one of the partner veterinary teaching hospitals every 6 months for an exam. And the primary endpoint for the rapamycin trial is lifespan. So we are powered to detect a 15% effect on lifespan. Secondary endpoints for that study include age-related cardiac function, age-related cognitive decline, activity, rates of age-related diseases like cancer and kidney disease and things like that. So we are following as many of the age-related changes as we can to ask, first of all, does rapamycin extend lifespan? And then secondly, does it delay the onset and progression of multiple components of the aging process?
Have you considered having a triad group with multiple compounds?
So, we certainly have considered it. Again, the limiting factor for all of these things, especially the triad study, is money, right? So we were very, I'm very pleased that we were able to get a large grant from the National Institute on Aging to do this, but it's not enough to do everything we would like to do.
Yeah.
So certainly there are other interventions you could imagine testing using a model very similar to the triad design. And then there are combinatorial approaches that you could envision taking. The one approach that we were not able to take that I really wish we had been able to take is a transient rapamycin treatment group. So in mice, we know that you can start treating at about 20 months of age, which is maybe the mouse equivalent of a 60-year-old person or a 7-8-year-old dog, and you get lifespan extension. And then my lab has shown you can actually treat them only for 3 months, from 20 to 23 months of age, and you still get pretty much the same lifespan extension and improvements in certain measures of health. So it would be, I think, extremely interesting and informative to know in dogs, if we only, instead of treating the cohort for the full 3 years, let's say we only treat them for 6 months. Would we see similar effects in that transient group compared to the continuous group? That actually was in the grant proposal, and the reviewers disagreed with my enthusiasm for doing that part of the study. So we didn't get that part of the study funded. So absolutely, there are lots of things that we could do. My hope is that by showing people that we can do this for rapamycin, this will be a proof of principle that will then allow perhaps us, perhaps other people to take similar approaches, with the template already there. And I think it's important to think, we've been talking about the triad trial in dogs for long enough now that I think a lot of people forget that, this is the first time anybody has ever even thought about trying to do something like this. And so it's a big undertaking. And when I first started talking about this, a lot of people thought I was crazy because they thought there's no way you can do a clinical trial for lifespan with rapamycin in dogs. Owners aren't going to do it. You'll never get funded to do it. So I think just showing that this is possible, that people want to participate, will be an important outcome of triad, independent of whether or not we see the effects of rapamycin that we anticipate seeing. It is a clinical trial. It might not work. I think we all have to accept that. But I think showing that it can be done will hopefully set the stage for for similar studies in the future, testing other interventions where we have reason to believe from our preclinical studies that they could have a positive effect.
Right.
It's important to prove examples.
Yes,
Yeah.
absolutely.
So in worms, when they would manipulate C. elegans, people would target a gene called DAF2, and it was they would trigger a hibernative mechanism in worms and cause them to extend their lifespan through a Dower phase. This is one of the things that can't get translated to humans because we don't have that, or it's not thought that it doesn't appear that we have the hibernative state that those worms have. Is there any core feature of dogs that has a similar limitation, I guess, if you're to try to translate from dogs to humans? Is there anything that you know of right now that is like, hey, here's something that's weird about dogs that's not going to be part of human biology?
Sure. So let me split this up into two answers, because I want to clarify the first part of this question, which is that the DAF2 example in worms wouldn't translate to people. You're right that mutations in DAF2 will cause worms to go into this Dower state, which is a developmental hibernating state. It's a foraging, a diapause. But what has been shown is that if you wait until the worms get past that branch point into Dower till they're just before adulthood, and if you knock down DAF2 then, you can still get the lifespan effects during adulthood. So the lifespan effect doesn't require the worms to go into Dower. It's just the same pathway that affects both Dower and lifespan. And in fact, mammals do have a much more complicated version of this pathway that DAF2 acts on. So in worms, it's called the insulin IGF-1-like signaling pathway. In mammals, we have insulin signaling and we have IGF-1 signaling. But there's good reason to believe that at least the IGF-1 pathway in particular is important for aging in mammals. So I think that at least some of what DAF2 does in worms to extend lifespan probably is conserved in mammals. And in mice, we know that's the case. And there's some reason to think it probably is true in people. And there's some reason to think it probably is true in dogs as well. So it turns out that if you look at the single greatest genetic predictor for body size in dogs, it's IGF-1, which is the pathway that DAF2 acts on. So dogs that have a genetic variant of the IGF-1 receptor that causes them to have lower IGF-1 signaling, which is what knocking down DAF2 does in worms, are smaller. And we know that smaller dogs live longer. So, we haven't proven the causal arrows that the IGF-1 variant is causing increased lifespan in dogs. But I think it's a likely hypothesis based on this conservation from worms. So I just wanted to make the point that this example you made, actually, there's some reason to think it might be relevant in dogs and in people. Okay, so then the second question, is there anything about dogs that makes them, maybe not a great model for human aging? And I think you can certainly point to differences. So one of the reasons why companion dogs are a good model potentially for human aging is they get most of the same age-related diseases that we do. They show the same functional declines in organs and tissues that humans do. So there's lots of reason at the clinical level to think that aging is similar between dogs and people. But of course, they do it much faster. They age much faster than people do, which sucks. As somebody who loves dogs, that sucks. But it does make them a very powerful animal in which to study the aging process. And I just want to take a second for an aside here because we're talking about the relevance of dogs for human aging. I want to make the point that the Dog Aging Project is not a project that's trying to study aging in dogs solely because of what it's going to tell us about aging in humans. That's important, but those of us who work on the Dog Aging Project all have dogs. We all love dogs, and we're driven by developing therapies that will enhance health and longevity in dogs. And I think there's intrinsic value in doing that, independent of what we're going to learn about human aging, which is also important. I just want to make the point that those are parallel goals that are not mutually exclusive, and both are important. Okay. So your question, is there anything about dogs that might suggest that they're not a perfect model for human aging? And I think you can certainly point to a few things. One is that most of the cancers, the types of cancers that dogs get with age, are actually more commonly pediatric cancers in people, which suggests some interesting biology. but we don't really understand it. So it's not exactly the same spectrum of age. So cancer is age-related in dogs, but it's not exactly the same types of cancer. The other area where I think there are obvious differences is diet, right? So very few dogs, fortunately perhaps, eat a diet that is substantially similar to the typical diet that people, at least in the United States, would eat. So that's a difference. But I think where dogs have a similar power is that the diet in dogs is variable. So just like human diets, there is not one diet that every dog gets. There are a variety of diets, and we, at least in principle, will be able to draw correlations between specific diets that dogs are receiving and their aging trajectories as they go throughout their lives. So that is an advantage over laboratory studies in mice, where while there are a few different diets that people feed mice in the lab, it's pretty similar from study to study and lab to lab, the composition of those diets. Dogs have a much more variable nutritional experience compared to laboratory animals. So it's not exactly the same as people in terms of the composition of the diet, but at least there is that variation. And I think in general, that's both a challenge and a power of the Dog Aging Project. That variation in environment is something we can't study in laboratory studies. And in fact, we intentionally try not to study that. We try to limit the environment and control it. In the Dog Aging Project, we intentionally try not to limit the environment. We want the environmental variation. We want the genetic variation, because that's the only way that we can really figure out what are the important factors that affect aging in the real world.
All right.
Oh, sorry. Let me say one more thing about dogs.
Okay.
That's different. So most people don't appreciate this, but there are actually 4 common sexes in dogs, right? There are intact males, intact females, sterile males, and sterile females. So that's another area that is different than the human population. But it's also, I think, potentially will lead us to some interesting biological insights, which is the importance of sex and sex hormone signaling on healthy outcomes during aging. And there's already some literature on this that show that intact dogs develop age-related diseases at somewhat different frequencies than sterile dogs. And so I think we'll have an opportunity to actually learn something from that difference as well.
Oh yeah, I didn't even think about that. That's interesting. Okay, so I think we're getting close to the end of the allotted time. And do you have any last questions, guys? Or okay. Well, your project is very interesting, and we'll be watching, see for developments. And we thank you for spending your time here today with us.
Pleasure.
And I guess that's the end of the call. And I wish you well in your endeavors, and hopefully you make a lot of progress.
All right. Thanks a lot, guys. Take care.
Thank you for your time.
Thank you.
Goodbye.